Good afternoon. My name is Renelle Miles, and welcome to NIH's Office of Disease Prevention Medicine Mind the Gap seminar series. This seminar series explores research design, measurement, intervention, data analysis, and other methods of interest to prevention science. Our goal is to engage the prevention research community in thought-provoking discussions to promote the use of the best available methods in prevention research and to support the development of better methods. Before we begin, I have some housekeeping items. To participate by Twitter, follow us at NIHPrevents and submit questions using the hashtag NIHMTG. You may also email questions to prevention@mail.nih.gov. There is also a link to a feedback form at the bottom of the videocast page where you can submit questions during the talk. At the conclusion of today's talk, we will open the floor to questions that have been submitted via Twitter and email. Lastly, we would appreciate your feedback about today's seminar. Following today's talk, please visit the ODP website at prevention.nih.gov/mindthegap and click the link to the seminar evaluation under the resources section. Your insights will help us continue to improve this seminar series. At this time, I'd like to turn things over to Dr. David M. Murray, Associate Director for Prevention and Director of the Office of Disease Prevention.
Thank you, Renelle. I'd like to introduce our speaker today. Dr. Todd Little is a professor and director of the Research, Evaluation, Measurement, and Statistics program at Texas Tech University and director of the Institute for Measurement, Methodology, Analysis, and Policy. He's internationally recognized for his quantitative work on various aspects of applied structural equation modeling, particularly modern missing data treatments, and he's known for his substantive developmental research in action control processes and motivation, coping, and self-regulation. In 2009, Dr. Little was elected president of the American Psychological Association's Division of Evaluation, Measurement, and Statistics. That's Division 5 for those of you in APA. Dr. Little is a fellow in the American Association for Psychological Science, the American Association for the Advancement of Science, and the American Psychological Association. He's published with colleagues from around the world in over 65 different journals, garnered over 17,000 citations for his work. He published a text, Longitudinal Structural Equation Modeling, in 2013, and has edited 5 other books related to methodology, including the Oxford Handbook of Quantitative Methods and the Guilford Handbook for Developmental Research Methods. Lastly, Dr. Little founded, organizes, and teaches in the internationally renowned STATS camp. So at this time, I would like to welcome Dr. Little and turn the session over to him. Dr. Little.
All right, thank you, David, very much for the kind introduction and for the invitation to be here. When we met at the P2P conference, I did mention that an opportunity to talk about missing data. I rarely pass that up, so I'm particularly pleased to be here. So what I want to do today is try to convince you that the modern treatments for missing data are useful for correcting problems that emerge, and basically to learn why not imputing your missing data is more likely to lead to errors in generalization. It's not cheating. It's not making up data. It's all these misconceptions I hope to dispel. And if I do my job right here today, by the time we get toward the end of my discussion, I hope to truly blow your mind with regard to some of the advantages that missing data can actually provide when we start planning for missing data. So the key issue here when we have missing data, of course, is recoverability. So can we get the sufficient statistics to look like what they should have been had we not had missing data? So we're looking at the means, the variances, and the covariances there. Or from another perspective, can we get the parameter estimates of any statistical model that we're fitting to the data to look like what they should have looked like had we not had missing data? Because when data go missing, they go missing for a reason. And the key consideration here is, can we recover that reason so that the parameter estimates, the statistical model we're fitting is unbiased? So the other consideration, of course, is bias. First of all, if we do nothing to treat the missing data and they've gone missing for a reason, we're going to have bias. Secondly, if we treat the missing data, is there any evidence that the particular treatment that we're choosing to rectify or to treat the missing data that might also lead to bias? Now, the principled modern approaches that I'm going to be talking about today, there is no evidence that they introduce bias, but there are some traditional approaches to treating missing data that exist prominently in prevention research, for example. When you're looking at clinical trials, the practice of using the last point carried forward is particularly deleterious with regard to inferences, and we really should be treating the missing data using a modern approach. The third major consideration when data go missing is power. When data go missing, we lose information, we lose power. So when we use our listwise or our pairwise deletion methods, we're doing nothing to recover the power. In fact, we're introducing even more power loss, and we're doing nothing to try to recover the reasons for the missing data. In that sense, too, by further leveraging deletion methods, we are still introducing potential further bias. So basically, the treatments that have been in vogue for many, many years generally are not going to do anything to help with these three considerations. The modern approaches to treating missing data, on the other hand, are going to help both rectify the problems that occurred when the data went missing for a reason, be as unbiased as possible, given that you have missing data in the first place, as well as bring back considerable amounts of the power loss, not 100%, but a considerable amount. I think to really appreciate then the modern approaches, we do have to delve into the mechanisms that give rise to missing data. There are three mechanisms that are in the literature. I did not name these. These are the names that have been existing for a while. The first mechanism, missing completely at random, MCAR, and I want you to emphasize the completely in the word in this mechanism, because really that means that there is no association with the observed variables. It's basically every data point has gone missing for a unique reason. It has no impact on the quality of the inferences. The only thing that we lose when the data are missing completely at random is we lose power. The only thing that we lose when the MCAR is the mechanism. Most of the traditional approaches assume MCAR is the only mechanism that's involved in the reason for missing data, when in fact it's a rare mechanism. It doesn't happen in nature that often. Most data go missing for a reason. That's and we need to then figure out which of the two possible reasons when they do go missing for a reason, what is happening here. So the missing at random mechanism, MAR, is when the data go missing for a reason, but I have that variable on my data set, and I'm able to utilize that information, leverage that information to rectify and condition my data so that now it's as if the data are missing at random, or it's a random predictable process, however you want to think about it. But missing at random means the data have gone missing for a reason, and if we treat it properly, we can get back that particular reasons or that reason or those reasons for why the data have gone missing. The third mechanism, the missing not at random or the non-random selective missing, MNAR or NMAR, depending on your flavor of dyslexia, the data have gone missing for a reason, but unfortunately I do not have a variable on my data set that represents that particular reason, and therefore I have nothing I can condition my data on to rectify the problem. The classic example here would be something like income. When you ask people how much money they make, typically people at the high end don't tell you how much money they're making. The variable that you need to know how much money they're making is the income itself, and it's missing. So we'd have no way to infer who is very, very rich because that information is not available on the data set. We would have a selective missing data mechanism then. On the other hand, if I'm thinking about missing data and the potential that some people will not respond to that particular item, I can ask questions that might be related to income. And so I can hopefully have variables that become proxies for income that would be assessed on my data set and that would therefore allow me to correct for and be able to leverage that information to basically convert the mechanism into a MAR mechanism. So what I want to do is move to the next slide to kind of demonstrate the relationships among these mechanisms. Now what you'll see in this particular slide is the missing completely at random mechanism is a truly random process, and that mechanism is uncorrelated with the MAR process, uncorrelated with the NMAR process, and doesn't predict anything about the missing data values. It's a perfectly benign mechanism. The MAR mechanism is a measured predictable process. And what you'll notice there is that the MAR mechanism will be correlated to some degree with the variables that are in the NMAR pool. So the unmeasured but potentially unpredictable process, as long as I can get that correlation in the lower right-hand part of the screen, that canonical relationship between variables that are on my data set versus the variables that are not on my data set, if I can get that canonical relationship to approach 1.0, then the information that is lost becomes completely ignorable. That is the relationship of the end of the MAR mechanism to the missing data. That canonical regression path there, which I have listed here as not zero, as it begins to approach 1.0, means to get back the reasons, I'm going to get back the power, and I'm going to minimize any potential bias that occurred when the data went missing. So the goal here is to come up with variables in your on your protocol that could be predictive of the missing data. And so I like to try to think to help people think about this, is to think about your missing data model as different from your analysis model. The missing data model is going to be those things that you anticipate measuring for the sole reason of recovering the missing data mechanisms. So to try to convert as much as possible any potential NMAR mechanism into a MAR mechanism, so that when you do fit your analysis model, that it will be as unbiased as possible. So for example, if income is the issue that you think is going to be missing and you want to make sure you recover it, you might ask people, do you own a second home? You might collect zip codes and say, oh, this person lives on Snob Knob, they're likely to be very, very, very rich. So we have variables that we could incorporate in our data collection protocol that would eventually begin to start converting the MNAR mechanism into a MAR mechanism, so that we would be able to recover the information that is lost. For those of you who might like formula, here are the probability formulas that describe the missing data mechanisms. The key element here, again, is we want to try to get to that MAR mechanism by having variables on our data set that when we condition for it, we do have the probability of the data given the missing data mechanism that is essentially converts it into an ignorable process. Now, one of the key things to keep in mind, I'm going to jump ahead a slide here if you don't mind. There are two basic modern data missing data approaches: the MI, or multiple imputation, and full information maximum likelihood. The two approaches have been around for some time, since the 1980s. Multiple imputation, the goal there is to recover the sufficient statistics. So multiple imputation will, in fact, place plausible values where there are missing data cells, with the goal that those plausible values are trying to recover the variances, the means, and the covariances among the variables that are existing on the data set. It's not trying to say something about the person, but it is trying to say something about the population from which the sample was drawn. So multiple imputation is a data-based approach that uses typically a Markov chain Monte Carlo mechanism, or perhaps the EM algorithm, but it's primarily going to be using more of a Bayesian approach to reproduce the sufficient statistics. Full information maximum likelihood, on the other hand, attempts to fit the analysis model in the presence of missing data, and it uses marginal likelihoods, weighting the cases that have more data get weighted more, but it still will recover the missing data mechanisms when the variables responsible for missingness are part of the analysis model. So let me go back a slide to slide 7 again. So what I wanted to emphasize here is that if I have a data set, a large data set that has many variables, and I'm fortunate enough that the variables responsible for missingness are on my data set, if I choose to use full information maximum likelihood, the key consideration there is that the variables responsible for missingness must be included in the analysis pool somehow. If they're not included in the analysis pool, then that information cannot inform the model parameters. The model parameters will not be corrected for bias. I'm basically going to be treating the data as if it was missing completely at random, which is not the case, so that my results would end up being biased. And I think one of the problems right now in the literature, when people utilize full information maximum likelihood, they will make a statement like, well, we assume that the data are missing at random, and therefore we have corrected for the bias, that's not true unless a variable or those variables responsible for missingness are included as part of the analysis model. Now, there is a way to do this. It's called using auxiliary variables. So a key consideration in the missing data literature is identifying and incorporating auxiliary variables into your analysis model that would inform the missing data model. So the auxiliary variables are there only for the sole purpose of informing the missing data mechanism, so that when you do your analyses, the analysis variables or the analysis model will be corrected for the biasing effects that occurred because the data went missing for a reason. Auxiliary variables are not predictors, they're not covariates, they simply are saturated correlates. They dangle in the model to inform the model's parameters when you do fit the analysis model that you're intending to fit. So these are the two modern approaches, FIML or MI. Both are equally good. Both will give you the same results, all things being equal. Multiple imputation, generally you would want to do about 100 imputations. So what MI does is it starts off with plausible values. It goes through an iterative process to come up with the most plausible, and it does that over and over and over again, up to 100 times, and then you summarize the information across the 100 imputations to get at what the model parameters should be based on fitting your analysis model to each of the 100 imputed data sets. FIML, on the other hand, is a one-time analysis system. It fits the data, fits your model to the data in the presence of missing data and makes the corrections based on the information that is part of your analysis model, as well as information that's contained in the auxiliary block. Now, when we do imputation, one of the things we do need to be mindful of is where are we trying to generalize? So, for example, if we're doing aging studies and we want to generalize to successful agers, we have people who have dropped out because they've died. We are not going to impute those missing data because they are not part of the sample that I want to generalize to. Skip and fill patterns, so that can't happen. We need to reset some of those values to missing if we use multiple imputation, for example. So if we have variables that are asking about pregnancy, the missing data mechanism will impute scores for the males. We would need to reset those values to be missing because they are not plausible. Now, I want to emphasize here the importance of the auxiliary variables. This is a simple little simulation where 60% of the data are missing, 60% are missing, and they're missing for a reason. I've just lopped off the lower part of the distribution, so the direction of the bias here is less important than the amount of bias. So here I have a simple simulation where I have an X variable, a Y variable. I've simulated the correlation to be 0.3. I've varied sample size from 50 to 1,000. So when 60% of 50 are missing, that's quite a few people missing. And we can see the 95 and 99% confidence interval is quite broad. And we have a biased correlation. The estimated correlation is around 0.55 in this example. Now, when I include auxiliary variables and recalculate the XY correlation, what we'll see is, in fact, I will get back the bias that occurred. The auxiliary variables inform the correlation. They're not, again, they're not predictors. They're just saturated correlates that allow the XY correlation to be estimated properly. And you'll also notice that the confidence bands, the 95 and 99% confidence interval, have shrunk considerably, such that I have the power that I lost using these 8 auxiliary variables that were part of this example. Now, one of the key issues with auxiliary variables is that we want to try to use all of the variables on our data set that are not part of the analysis model to be included as auxiliary variables. This is called the inclusive strategy. And a key problem with this strategy, however, is that the auxiliary variables themselves will often have missing data, and it will put a challenge on the estimator such that once you get more than, say, 30 or 40 variables involved in the imputation process, there are too many patterns of missing data, and both MI and FIML will often not converge. That you've pushed the capacity of the software to its breaking point. We've introduced a method called principal components auxiliary variables, which is a method by which we take the information that's contained in the data set and distill it down into a meaningful set of principal components that can then be used as auxiliary variables. So in this particular example, on the next slide, I've converted the 8 auxiliary variables into a single principal component and refit the analysis model using that one linear combination among the 8 auxiliary variables, and we see that we get back the same recoverability. We get back, in fact, we get back even a little bit more power recovery using the single principal component as opposed to the auxiliary variables, which themselves are correlated. And as we see on the next slide, that when the auxiliary variables have about a 0.4 correlation among each other, we start getting into collinearity problems such that we don't get as much power back with a saturated auxiliary variable approach as we do when we do the principal components approach. So the one principal component auxiliary variable actually does a better job when there is collinearity among your auxiliary variable set. The approach that we're recommending using the principal components approach, what it does is it takes advantage of a program that we've developed here. I just wanted to show the convergence problem with the PCA auxiliary. You can just see that the iteration chain is much crisper and more reliable as it gets to the point with one PCA versus the auxiliary variables. But what I mentioned here and the next is there are some macros available to help you with the PCA auxiliary variables. One of them in particular is a routine that we've developed called Quark. My students named it. I have no idea why they've chosen that, but it is what it is. It's called Quark, and it's in the R package. It's a free open source package. Right now, it is not on the CRAN, but if you go to camp.org, you can go to materials and you'll see where you can download Quark. What Quark does is it's an automated process by which it can take extremely large data sets and distill them down into a meaningful set of auxiliary variables that you can then use in your analysis or in your multiple imputation approach. So you can use them either for multiple imputation in a more sequential manner, because again, even with multiple imputation, if I have lots of variables with lots of patterns and missing data, the MI will not converge. But with the auxiliary variables, I can actually do what's called isolated imputation. I can take it sequentially. I can grab a one or a set of variables, impute them in the context of the auxiliary variables that I've extracted from the data set, save those imputations, impute another set, and then merge by imputation and ID such that I can get back a completely imputed data set for using for any analysis purpose. Right. So with what I've described thus far are techniques for dealing with unplanned missing data. What I want to take is the next few minutes of this presentation is I do want to talk about a couple of planned missing data designs that leverage the power of the modern approaches for treating missing data. So if we know that we're going to have missing data problems that occur in nature, and we've done everything we can to plan for it in terms of including auxiliary variables, we know that we're going to be using FIML or multiple imputation. We know we're going to be running the Quark package to get the principal component auxiliary variables. We can now also incorporate on top of that planned missing data such that we as the investigators are going to control where and how we have missing data. We're going to utilize the missing completely at random mechanism. And there are a couple different techniques, planned missing data techniques that we're going to talk about. I'm going to talk here briefly about the multiform protocol. Now, the multiform planned missing data protocol, basically it takes long assessments that often reduce data quality. Those of you who have recently filled out a questionnaire quite often, if me, you lose your attention halfway into it, and you might even stop responding before you get to the end of the questionnaire. This is something that is human nature. It's something that's ubiquitous and universal. One of the things we want to try to do is to reduce that kind of unplanned missing data. We can get repeated assessments that can induce practice effects. Planned missing data designs can actually be utilized to estimate and control for practice effects. We can take data collections that are time and cost intensive, and we can reduce the amount of time and the amount of costs that are involved, which for the panels that are spending, the NIH panels that are trying to save money on their data collections, planned missing data designs are one way to do this to save costs and yet and keep the power and the generalizability to where we would want it to be. So the multi-form design, I'm going to talk about the three-form design in particular with one quick extension. I'm also going to talk about the two-method plan missing data design. So for the multi-form design, there are a couple, here's two flavors of the three-form design. The way that this three-form design and all multi-form designs work is you identify some variables that are going to be given to all participants, and those are referred to as the X set. So the demographic variables that are going to be critical, the gating variables that you might need to know about, those are good candidates to put into the X set. For those constructs that have multiple indicators, those are great candidates to start taking those items and to spread them across variable sets A, B, and C. So what you see here is we have 4 different variable sets, X, A, B, and C. And what we're going to do is we're going to create 3 different short forms, but 3 different short forms that are comprised of sets of variables that allow for all possible variables to correlate with all other variables, such that we can infer what the relationships between all variables should be. And if we then take each form and randomly assign participant to form, the reason for the missing data is now missing completely at random. So for the top part of this, I've said, let's say maybe take about 1/4 of the items and put them into the X set, and 1/4 into the A, and 1/4 into the B. What that means is I would be creating about 1/4 of my variables would be missing for each form, because form 1 has X, A, and B items, but not the C items. Form 2 has X, A, and C, but not B. And form 3 has X, B, and C, but not A. So each form is missing about 1/4 of the items. Those are missing completely at random. On the bottom side, I've created a more aggressive version of the multi-form design where I'm putting about 1/3 of the items into variable sets A, B, and C, so that about 1/3 of the items are not being presented by utilizing this missing completely at random mechanism, by assigning form to person. So as a quick example, let's see here. I'm sorry, this slide, the two-method design is out of place. Sorry, I'll put that back. Three-form design. So here's an example where I have questions made up of with demographics. I'm looking at musical taste. I have the big 5. So what I'm going to do is I'm going to put the demographics, the musical taste. They go into the X set. I take the first item from each of the big 5 and assign them to variable set A. Take the next and assign them to variable set B, and the next to variable set C. And here then I create forms. Form 1 has the X, A, and B items. Form 2 has X, A, and C. And Form 3 has X, B, and C. When I put them back together, my data set, my data frame, has just as many columns as I would have had I done a complete case or a complete protocol design, but I now have these holes poked into my data set that are actually holes created by me by randomly assigning person and form, so that these missing cells, these missing elements, are missing completely at random. Now, the multi-form designs will reduce the unplanned missing data as well. So, for example, in this case, I could have unplanned missing data on top of this, but it's still going to be minimized because I've got a much shorter protocol. People are going to give me more attention as these protocols are being delivered. Okay? So once I go through the imputation process, I would bring back the power, and I would have the ability to generalize back to the population from which the sample was drawn. In terms of sample size considerations, the minimum sample size for the three-form design or the multi-form design is about 40 persons per form, or a sample size of about 120 in order to implement a multi-form design. If your sample sizes are going to be smaller, you need to put more into the X set, so you're introducing less aggressive forms of missing data. And if you have larger sample sizes, you can go more aggressively. For example, here is a 10-form 6-variable set variation of the multi-form design, where I have an X set that everybody gets. Everybody gets asked the X items. But then I have some items that I've assigned to A, some items to B, some items to C, some items to D, some items to E. Generally, they're going to be about equal, but they don't have to be. We generally want to try to get the variables across, or the between sets variables to be highly correlated. So I like to take items that are highly correlated with each other and split them into different variable sets. Items that are measuring the same construct, spread them out across the different variable sets. And what you'll see, for example, is one of the forms that I would create would be the X items plus the A items plus the B items. So I would have X, A, and B. I would know how A items correlate with B items. In form 2, I would have X, A, and C. So I would know how the A items correlate with the C items. In form 3, I would have X, A, and D. In form 4, I have X, A, and E. And then moving on for form 5, I now pair the B items with the C items, the B items with the D items, the B items with the E items, and I continue on until to get all possible pairs of items to be presented together. I now have 10 forms, and I would take these 10 forms and I would randomly assign person to form, again with a sample size of about 40 persons per form. This would mean I would need a sample size of about 400 to implement the 10 form plan missing data design. But what you can see here is in an aggressive use of this design is about anywhere from 3/5 to 50% of my data are missing completely at random. And using practices like Quark or Fimmel estimation with auxiliary variables, we would get back a complete frame data set that would allow us to have perfectly valid inferences based on this multi-form plan missing data design. All right, now the last design that I want to talk about before we get to the question and answer period is the two-method plan missing data design. Now the two-method plan missing data design is actually a method where we can increase power by leveraging what we know about both missing data as well as latent variable modeling. So the way that the two-method plan missing data design works is that there's typically going to be, it works when there are two methods for measuring the same underlying construct. So we have what's called the expensive measure, which is the gold standard. It's highly valid, it's unbiased. The problem is it's time-consuming and quite costly, and it's not feasible to collect from a large sample size. So if we look at biological assays, or if we look at anything done like done under a magnet, or in doing an intelligence assessment, or anything that is very expensive, can often be both costly and time-consuming. And what happens when folks want to start examining more nuanced aspects, large sample aspects of what happens with if I'm relying only on the gold standard, I'm typically only going to be asking main effect, big effect questions. I can't ask nuanced questions about unique effects, mechanisms of change, those kinds of things. So the folks that are interested in mechanisms of change often will resort to developing an inexpensive, cheap measure, but it has bias. So it's a self-report measure of stress, or it's a self-report or a teacher report, or some other thing that I can get from a lot of people, but the problem is it's going to be biased, but I can get it from a large sample. Now, if I can, here's a couple of examples. If I look at stress, the expensive measure might be to do the cortisol assays, or I could survey stressful thoughts. I can look at intelligence. I can do the WACE IQ. I could create a simple multiple choice. I could look at smoking, student attention, anything that has potentially two methods for measuring it. I have an expensive measure that is costly and an inexpensive measure that is easy to assess. And what I'm going to do is I'm going to give the inexpensive measure to everybody. So I'm going to select a sample maybe around 120, 140, so that I can start looking at some possible unique mechanisms of change, and then I'm going to randomly assign about 1/3 of the sample to receive the expensive measure. So 1/3 will get the expensive measure, but everybody will get the inexpensive measure. And when I do that, then I can fit a latent variable model like the one I've got depicted here, where I have the construct of interest. In this case, on the left-hand panel, I have smoking cessation as the example. It's anchored in the blood draw, so I know from the CO2 and cotinine information from the blood draw the amount of smoking that occurred in the prior period, but I also then use the an inexpensive measure where I ask people to self-report on their smoking behavior. And I might have multiple indicators of that particular smoking, asking about, at workplace, at home, morning, afternoon, querying smoking in numerous instances. So I have multiple indicators of self-report smoking. I have multiple indicators of actual smoking based on the blood draw. The data are missing completely at random because I've randomly assigned who was going to be giving me the blood information. I didn't let them know beforehand because that would change their response pattern. But now what I have is an opportunity to extract a latent factor, the smoking cessation that is unbiased, that is anchored in the blood draw, that is a factor score for all individuals in my sample. So that is an unbiased construct that and what makes it unbiased is by stating a bias-factor model. So you see in this particular model, I have a second construct, which is extracting the bias that occurs from the self-report information. That's what makes the smoking cessation unbiased construct unbiased, is by extracting that the self-report bias in that measure. But more importantly, that particular construct also contains behavioral information about the degree to which you are willing to lie to me. It is a social desirability, a behavioral aspect of social desirability that we can actually now utilize as a control construct in this particular analysis model. So let's say this was an intervention trial that was looking at things like efficacy for smoking, attitudes about smoking, both of which might also have some self-report bias or some social desirability in them. And if I wanted to see how those constructs actually relate to smoking cessation, I can actually take this bias factor that I've created by using the two-method plan missing data design, and I can now use that bias construct as a control construct to extract the amount of social desirability from those other constructs to actually see what effect they might have and their importance in the intervention that I'm trying to examine. So the two-method plan missing data design is actually a way to leverage power where it didn't exist otherwise. If I was limited in resources, I would have had a sample size of maybe 40 or 50 individuals where I did the blood draw, but I would not be able to do any interesting latent variable SEM analyses. I certainly couldn't do a longitudinal model where I'm looking at mediation processes or other mechanisms or any moderators or anything like that because I'm underpowered. But by utilizing the the two-method plan missing data design, I can actually get back a sample size that has significant power as well as an unbiased estimate of the key constructs of interest, in this case, smoking cessation or actual stress. And with that, I think we will pause here and open questions. And I think I kept to my 30-minute timeline.
Yes. Thank you, Dr. Little, very much. Appreciate the presentation, and there is plenty of time for questions. The material that you presented was very interesting to me, much of it familiar, but some of it not. I'm intrigued particularly by the two sections that you presented in the sort of the second half of your presentation, the multiple form design, and then the two-method plan missing design. And I had some questions about those. So the multiple form design seems to be a lovely way to shrink the size of what otherwise might be a very lengthy survey instrument and use MCAR as a basis for getting back answers on all items, even the ones that you didn't ask. And if you have the shorter item, you may have less missing data generally because people are able to get through the whole thing in a shorter time rather than not getting to the last part of the questionnaire. So all of that seems quite positive. So what's the downside? I'm trying to figure out what am I risking, or what are the chances that I'm taking? What are the assumptions that we're making? Is there a downside here?
Yeah, David, that's an excellent question. The downside generally is it's a little bit more upfront work to create the multiple forms to ensure that you get your all variables are paired with every other variable just to make sure that your forms are properly constructed and that you use a truly random assignment process of assigning individual to form. The other downside used to be that with these multi-form designs, if you taxed the multiple imputation procedure, or if you taxed the FIML estimator, that sometimes you would get, again, too many patterns of missing data, even with MCAR trying to minimize those patterns of missing data, so that the imputation process would sometimes be hindered. One of the things that I introduced was this package called Quark, which now allows us to do isolated imputation, so that I can always execute a multiple imputation that will successfully manage to deal even extremely large data sets. So I think the biggest hindrance right now is just going through and trusting the integrity of the statistical theory behind all of this, and implementing the design, because we do have that. The fundamental problem in academia is there's risk aversion, and trying something new like a planned missing data design it feels risky. But the techniques themselves have an unequivocal statistical underpinning, and just kind of going through the process of learning how to properly execute them. I think once we have more and more people with that success experience of putting them together properly and seeing how effective they can be, then we'll start seeing them become more the matter of course rather than the exception.
Right. One of the questions that people ask me all the time is, David, if I propose this new method as part of my grant application, are the reviewers going to accept it? Because they seem to be averse to novelty when it comes to methods, even though they're supposed to be submitting things that are innovative. Or have you had experience with submitting applications, or are you familiar with applications that have gone to NIH using this approach that have gotten through the review process successfully?
Yes. In fact, we have now a great track record of proposing these techniques to NIH, IES, NSF, all of which I think that part of that is because, you have to make your case carefully, but there's also now a rather extensive literature out there that you can cite to support your arguments for implementing these designs. And I think that people are beginning, and if you have somebody on the panel that is reasonably astute methodologically, they're going to be aware of these designs, and they're going to be there to help make the argument in favor of the design when maybe some of the review panel may not be familiar with it.
Right.
But we've had a great deal of success utilizing these planned missing data designs as part of our protocols, and they are getting funded. And what's nice now too is that they're being the results are being published in our top journals, so we're actually seeing them being implemented more, so that it's something that people are becoming more familiar with.
And do you have a description or some text material that you can make available to interested parties, perhaps available on your website or just through an email solicitation, that people could use that describe this and document some of the history of its use in other projects?
Yeah, I think we could put something together. I could basically look at one of the latest grant proposals we've submitted and take the section on missing data out and put that up on the web as an example.
Yeah, I can see how that could be of help to many others. It might eat into your consulting income, so we have to be careful there. But I think it could be helpful to other investigators, and if you're willing to share that, that would be great. I have a question related to the two-method plan missing data design. As I was listening to it, it struck me that it's another way of approaching a problem that I have approached from a different angle for decades. Imagine we're working on a study where obesity or body composition is our primary outcome. We're interested in perhaps percent body fat as the primary outcome. And the usual best measure for that is doubly labeled water. That's a very expensive measure to take. We usually can't afford to do it on very many people. It's messy. The consent process is a little bit more complicated than getting people to fill out questionnaires. And what we've tended to do in a variety of studies that I've worked on is do a pilot first, where we have a sample that's something like the population we're going to work in. We collect data based on doubly labeled water. We also collect lots of anthropomorphic measures of skin folds and other indications of body fat. And then we develop a prediction equation, predicting percent body fat as measured by doubly labeled water, using all of these inexpensive circumference and skin fold measures and perhaps in bioelectric impedance measures. And at the end of that pilot study, we can show that we can explain 85% of the variability in the doubly labeled water measure using these predictors. Then in the full study, where we're going to measure thousands of people, we only measure the predictors. We don't measure doubly labeled water in anybody. And we use a predicted percent body fat as the primary outcome for the trial. And there are a number of studies that NIH has funded over the years that have used that kind of approach. The method that you're talking about would collect the doubly labeled water measure in some of the participants in the real trial and the predictors from everybody, and then use missing data methods to get back the doubly labeled water measure for everyone in the real trial. I know that you haven't had much time to think about this particular example, but you may have considered this kind of situation in other contexts. What are the pros and cons of those two? In the method I've been using, I didn't have to use the expensive measure in many people, and I got a pretty good prediction equation that I could apply to everybody. In the method that you're talking about, you would have to use that expensive method in more people, so you're going to pay more for it, and deal with the consent issues and other kinds of things. But there may be some advantages. So if you can say something about the pros and cons of those two approaches, that would be great.
Sure. You're right. I haven't had a chance to think this through thoroughly, but my hunch is that the predictive equations you're getting is about 85% accuracy. What we would be getting with the two-method plan missing data design is 100% accuracy. So if that 15% is important to you, then you would have a more accurate direct assessment using the latent variable two-method plan missing data design, because it will be 100% correct estimate based on the gold standard, because that is now what's anchoring the latent factor score. Plus, you would be correcting your estimates for unreliability. So there's the disattenuating influences of unreliability that we are able to recover using the latent variable approach, using the two-method plan missing data design. The number of people you might need may not be that much more than what you did in your pilot study when you were developing the relationship between the two different measures. There's a paper by John Graham that basically says the more highly correlated the cheap measure is with the expensive measure, and in your case it's actually going to be reasonably highly correlated, that the amount of bias that you need to correct isn't as pronounced as, say, a self-report of stress versus actual cortisol. So the more highly correlated the two are, the fewer you need of the expensive measure to anchor it and to do the bias correction that occurs.
All right. Thank you. That's very helpful. One of the other ideas that you've introduced in this presentation that may not be familiar to some in the audience is this principal components approach to distill the number of variables that are being used either in the auxiliary set or more generally for the missing data estimation down to a more limited number where perhaps there's no missing data in those variables by virtue of that first step, and less correlation among them because they're principal components rather than variables that are just naturally correlated. I want to ask the same kind of question that I asked initially with the multiple form approach. What are the assumptions that we're making if we do this? What are the risks that we're taking? What do we need to address in the caveats or in the grant application where we're defending this and arguing that we're not taking too much of a chance?
Sure. Well, we do have a paper that's been published in Multivariate Behavioral Research, Waylon Howard, Micah Remtala, and myself. So there is a paper out there that introduces the approach. The basic assumptions are basically the same assumptions you would make even at your analysis level model. What Quark can handle, it can handle categorical data, it can handle ordinal categorical data, it can handle continuous data. It just basically recasts everything, goes through a process of doing a single imputation as it extracts the linear component scores. One of the nice features about Quark is it could also allow for the fact that the missing data mechanism could potentially be nonlinear, and it actually extracts nonlinear component scores to represent even more aggressively the potential missing data mechanism. The downside is, if you've got missing data and you don't treat it with one of the modern approaches, you're not going to get back the bias that occurred, and you're going to be underpowered. So here's a situation where we've introduced an approach that really doesn't have much of a downside once you implement it properly and tell it which variables are categorical and which ones are not, and then it'll go through the imputation process. And you need to know a little bit about the different imputation procedures to know whether you want to use, for example, predictive mean matching or do one of the other algorithms that are available. But that's just consulting with a knowledgeable expert about getting some recommendations for which ones to use in which circumstances.
Okay.
But it's actually a pretty from my perspective, I think once we get all of the kinks of Quark worked out and get that available as a routine, it's going to be one of those things that people will just routinely start utilizing because it's going to be that effective in terms of being able to recover the missing data.
Yeah, you said it was available as an R package. Is it available in SAS or SPSS or Stata? Any of the? At this point,
it is not. We've been developing it in the R world because it's free and open source. And so one of the things you may have to do is, if you're like Stata or SAS, is pop over to R, run the Quark routine, and then send the data back into SAS, which is something we routinely do that for folks at IMAP. Is they will give us their data with massive missing data problems, and we'll run it through Quark and then give it back to them in a form that they wanted to have back in SPSS, SAS, and so on. So we've had actually a couple large contracts, one with the Navy looking at the Millennium Cohort Study, and another one with the CDC looking at the Dating Matters project, for example.
All right, thank you. Dr. Miles has a question.
Yeah, and kind of related to what you were just talking about, I guess one of the participants saw on slide 15, it looks like a SAS macro to them, but you're saying it's R. She said
there is a SAS macro that we started to work on that is now behind the R package. We're hoping eventually that we could get that SAS macro as powerful and as capable as the R one, but we've spent more of our time developing the R package. Once we get the kinks worked out of that, then we could start thinking about developing it in SAS, Stata, and other potential packages.
And she wanted to know if you could briefly describe what it does.
Sure. Okay. So what Quark will do is you'll tell Quark what variables your identification variables are, your ID variable. You'll tell it which variables are categorical, and then if you don't, or which ones are like on a Likert scale, if you use something like a 7-point Polytomist or Likert scale, and then it assumes the other variables are going to be continuous. And and then the third component one is to tell it some potential grouping variables. Because what Quark will try to do is it'll try to do a single imputation, generally starting with something like an EM algorithm, then it'll move to predictive mean matching, and then it'll move on to group mean substitution based on your grouping variables until every variable has at least been successfully a single imputation. Then what it does is it extracts a number of component scores from your data set. The number that you request is part of the R package. We default to 10 component scores, which appears to be enough to capture more missing data mechanisms. The next step is to then actually to recast the remaining variables as nonlinear components. So we take product terms, powered polynomials, and the like, extract all the linear information out of there so that there's no collinearity there. But then from the remaining information, we'll extract 3 or 4 more nonlinear component scores, so that what you get back in the end is a data set that has your original data with the missing data plus 13 or 14 component scores on the data set that can be used as auxiliary variables when you do FIML estimation, or you can run it through a multiple imputation process and generate 100 imputed data sets, imputing against those component scores. And Quark automates all of that so that it's fairly straightforward to implement.
Okay, and I have a two-part question. What method do you recommend to identify predictors of missingness? And if an auxiliary PCA is not a predictor, how and where is it entered in a GLM type model?
Okay, so one of the things about the PCA approach is that it's agnostic as to which variable is MAR, but it's going to capture MAR if MAR exists on your data. Essentially, if the missing data is predictable, the component scores capture it. And so we can be kind of agnostic about that. We don't have to know which variables they were per se, although I do like to have people do an analysis of the missing data, and it can just be a simple logistic regression where you predict missingness from the variables on your data set and identify variables that seem to be predicting it so that you could inform people that, hey, we've got a MAR mechanism, these are the variables that were predictive of it. But once you move and recast them as auxiliary variables using the principal components approach, then we don't know, we just know that that variable is somewhere represented in those component scores. The component scores themselves are uncorrelated with each other. Now, if you're going to do a general linear model regression, for example, you may want to do a multiple imputation and then run your GLM 100 times and get the average parameter estimate from your 100 regression models. And SAS does this very easily, SPSS does this easily, Stata does this easily. Most of the software packages can easily do multiple imputation. You just have to tell it what is the sorting variable by the imputation number, and it will then use Rubin's rules to combine the parameter estimates after imputation. So the component scores are included in the imputation process. If you're using full information maximum likelihood, say a package like M+, you just have to, there's a statement in M+ that says auxiliaries are, and then you put PCAux1 through PCAux14, and you've got your auxiliary variables listed in the auxiliary block. And what M+ does is then it just creates a saturated correlate relationship of those auxiliary variables with the parameters of the model that you're trying to fit, and your model parameters will then be corrected for the missing data mechanism.
Did we get the second part of the question covered?
I think it was about the auxiliary PCA. If it's not a predictor, how and where is it entered into a GLM type model?
Yeah, so again, there you would have to do multiple imputation or use simul estimation. When you use simul estimation, there's typically an option to tell it what the auxiliary variables are. And so they're generally introduced as saturated correlates. Now, if you don't have that option, you can actually create the saturated correlates. You would enter them as variables, but they would just correlate with both your dependent variable and your independent variable. So you're going to have, some packages don't have that capacity, but if you were doing it in say a structural equation modeling packages to do your regressions, then you could create the saturated correlates where the auxiliary variables would just simply correlate with both the dependent variable and the independent variables.
Okay. Thank you.
Thank you for your presentation and all of useful information that you provided us today. And I want to thank everyone who participated in today's webinar. On the Medicine Mind the Gap website, which is again prevention.nih.gov/mindthegap, you will find several resources for this talk, including the slides, references, and a link to complete an evaluation. Your feedback is very important to us as we plan future sessions. Thank you again for your time.